PCR, or polymerase chain reaction, is like having a molecular photocopying machine right in the lab. Imagine being able to take a tiny piece of DNA and make millions of copies of it in just a few hours. That's the magic of PCR. Let's break it down. The process starts with a DNA template, which is the original strand we want to copy. To this, we add primers. These are short sequences of nucleotides that are designed to bind to specific parts of the DNA. Think of them as the starting points for our photocopying process. Then we introduce Taq polymerase, an enzyme that plays a crucial role in copying the DNA. Finally, we add nucleotides, the building blocks of DNA, which will be used to create the new strands. Now, on to the three main steps of PCR: denaturation, annealing, and extension. First up is denaturation. This is where we heat the mixture to around 95 degrees Celsius. This high temperature causes the double-stranded DNA to unwind and separate into two single strands. It's like opening a book to read it. Next comes annealing. We cool the mixture down to about 50 to 65 degrees Celsius. During this step, the primers attach themselves to the complementary sequences on the single-stranded DNA. This is crucial because it sets the stage for the next step. Then we move on to extension. We raise the temperature to about 72 degrees Celsius, which is the optimal temperature for Taq polymerase to work. This enzyme starts adding nucleotides to the primers, effectively building new strands of DNA. After this step, we've doubled the amount of DNA we started with. This cycle of denaturation, annealing, and extension is repeated multiple times, usually around 25 to 35 cycles. With each cycle, the amount of DNA doubles, leading to exponential amplification. It's incredible how something so small can be multiplied into millions of copies in just a few hours. The applications of PCR are vast and fascinating. In forensics, it's used to analyze tiny samples of DNA found at crime scenes, helping to identify suspects or victims. In medical diagnostics, PCR can detect pathogens, allowing for early diagnosis of diseases. And in genetic research, it's a fundamental tool for cloning genes, studying genetic mutations, and even in developing gene therapies. Sindel's amazement at the power of PCR is completely justified. This technique has revolutionized molecular biology, making it possible to explore the genetic blueprint of life in ways we never thought possible. It's a perfect example of how science can unlock the mysteries of our world, one DNA strand at a time.